In Chapter 13, we begin our discussion of the respiratory system. We use the term respiration in two different ways in physiology. One way that we use it is to describe the process of cellular respiration. And of course, we went over that earlier in our class. You're familiar with that backwards and forwards at this point in your biology career. That's not the way that we're using the term respiration, of course, in this particular chapter. The term respiration is also used to describe the exchange of oxygen and carbon dioxide with an organism's environment. And in this particular case, that's the meaning of respiration that we are using in this particular chapter, Chapter 13. So we begin with a discussion of the organization of the respiratory system, a bit of a review from anatomy. We are aware of the fact that the respiratory system consists primarily of left and right lung. Each of those lungs consists mainly of these tiny microscopic sacs known as alveoli. In fact, if we were to look at these alveoli and count them, there would be about 300 million of these per lung. And if we were to do something that couldn't really be done, but were to take these 300 million little tiny microscopic sacs out, cut them open and glue them together, we would see that these 300 million alveoli give us the surface area of approximately a tennis court for gas exchange. And so it's really an incredible adaptation. If we look at some animals that we would consider more primitive, like some of our amphibians, the plethodons, salamanders, for instance, they don't even have lungs. And so they are cold and slimy and can obtain all the oxygen they need across their skin. If we look at insects, they, of course, have the system of tubes in their body that takes oxygen from outside to deep inside their body. But none of those are nearly as effective as the lungs that we see in mammals and birds, and reptiles to a lesser extent. Amphibian lungs exist, but they are a little more primitive than mammalian lungs. So the overall purpose of the respiratory system is to bring air in contact with blood. The term respiratory cycle refers to one inhalation and one exhalation. And we go through several respiratory cycles per minute. And what we notice is that in resting conditions, we pull in about 4 liters of air per minute, and about 5 liters of blood flows through pulmonary circulation, therefore through the capillaries that we see in the lower right-hand side of our screen. So 5 liters of blood flows through those capillaries per minute, and that gas exchange occurs between the blood and the air. That's the whole purpose of the lungs, of course. What we see is that during exercise, depending, of course, upon a person's conditioning, the amount of air going into the lungs can speed up dramatically. So a 20-fold increase during exercise would not be uncommon. Whereas the blood, as we talked about in the previous chapter, you can increase, but you're not going to see a 20-fold increase. So a 5 to 6-fold increase would be expected during exercise when you're looking at the blood going through pulmonary circulation. Now, let's go ahead and take a look at the overall anatomy of this system, and we'll do so by looking at this diagram that's located right here in our notes. And what we see, and that's not entirely true. We'll use this diagram, and I'm pointing with the cursor here, if you hopefully can see. We'll use also the diagram in the upper right-hand corner of our notes. So air is inhaled, of course, via either the nasal cavity or the mouth, or both, and it makes its way into the pharynx. The pharynx is a passageway that's shared by both food and air. We have a split located right here, just before the so-called larynx, and the esophagus is located behind the trachea. The trachea lies in the front there. The trachea, of course, is a rigid tube supported by cartilage that obviously never collapses. The esophagus is a tube that is collapsed typically and makes its way down to the stomach. And so we diverge here between the digestive system and the respiratory system. We move now to the larynx. The larynx is the first part of the respiratory system that only is utilized by the respiratory system. We see that this is where the vocal cords are housed. We move on down into the actual remainder of the trachea itself. And then what we see is that we split. We have this splitting of the trachea into two bronchi. And the bronchi are the first tubes. One's going to go to the right lung, one is going to go to the left lung. And then there are 20 more branches after that until we get down to the very smallest branches of our respiratory system. This is often referred to as a bronchial tree, and we don't have any pictures of this in our physiology book, but you probably remember your anatomy text and the dissected bronchial tree that you saw, and it does look much like a tree. It has that dendritic connection of tubes that very much is reminiscent of the branches that you see on a tree. So we talk about just the main, we don't talk about all 20 of these, but we talk about the main categories. So we're going to shift our focus down to this diagram that's found in the lower middle of our slide. And so we're now, we've gone from the trachea to two bronchi, and then we have the number of tubes that are at each level over here on the right-hand side. We eventually make our way down into smaller and smaller branches, and we call them bronchioles. And so these are just very small tubes that have branched from the trachea. And then we have even smaller tubes referred to as terminal bronchioles. The terminal bronchioles are the last part of the respiratory system in which no gas exchange occurs. Everything up to this point has been for the conduction of gases, not for the exchange of gases with the environment. So now, and all of this is referred to as the conducting zone appropriately. So we see that labeled on the left-hand side of our diagram. So at this point, we shift over to the respiratory bronchioles. And so the respiratory bronchioles are the first part of this conducting system that have alveoli. And so alveoli are these little sacs that we mentioned previously where gas exchange does occur. And so we see a few on the walls of these respiratory bronchioles. Then eventually we get smaller and smaller. We make our way to alveolar ducts. And in the alveolar ducts, we have numerous alveoli that are found there. And eventually we terminate in these alveolar sacs. And so that's what's being depicted over here in this particular diagram. And in fact, on this slide, we can zoom in on it. I just enlarged this particular figure from our text. And so we see a bronchiole, and we notice the smooth muscle that is found around all of these structures once we get to small enough branches there that can constrict and perform different functions. One of the main functions that is accomplished by the constriction here is to divert air into places where there's more blood in the lungs. And so the lungs, as we'll see over the next little bit in this chapter, do a very good job of matching airflow and blood flow. And so that's partially accomplished by these smooth muscles. Here we have our terminal bronchiole that we mentioned previously. Again, no alveoli, so no gas exchange occurring. Here we have our respiratory bronchiole, so we do see the alveoli in the walls there. And then here we have our alveolar duct and our alveolar sacs. And so this is where it all ends, these little microscopic sacs, 300 million of these, not the whole alveolar sac, but the alveoli themselves. There's 300 million alveoli per lung. And so this is why, a consideration of what you take into your lungs, is certainly worthwhile. Imagine these 300 million little sacs. You certainly don't want things getting down into your lungs that your lungs can't get out. Your lungs are pretty good at getting things out, as we'll mention here. But it is possible for very small items, asbestos, for instance, to make their way deep into their lungs and embed there and not get out. So we now look at, before we leave this slide, let's look at this alveolus that we see here. So we see the, maybe we should go back over here. So here we see the blood coming in. Here's our pulmonary arteriole containing blue blood, which represents deoxygenated blood. And then we have pulmonary arteriole following it all the way out here. Here's our pulmonary arteriole, and it's delivering blood to the pulmonary capillaries. And so we can see that as the blood enters these pulmonary capillaries, around the alveolus. It's deoxygenated. The brightening of the color to red indicates picking up oxygen. Now, the pulmonary veins, of course, carry oxygenated blood, and so this is a pulmonary venule, but it's going to merge to a pulmonary vein off this diagram, it appears, and that's going to carry blood, of course, back to the heart to be pumped back through systemic circulation. We mentioned the conducting zone and the respiratory zone previously. The conducting zone, of course, begins at the top of the trachea and goes all the way down to the respiratory bronchioles, which are the first ones that contain alveoli, and therefore the first point at which gas exchange occurs. And then the respiratory zone is below that, the respiratory zone being, of course, the point at which, the area at which there's gas exchange that's taking place. One thing that we want to mention is this. We have, our lungs are pretty good at getting things out of them, and which should make us feel better about this topic. But what we see is that there are cilia that are present in our upper respiratory tract all the way through the conducting zone, even down into the respiratory bronchioles just a bit. And so these cilia, although they're not depicted on this particular slide, beat upwards, and they move mucus along the surface of those tubes that are, of course, lined by epithelial cells. And this mucus continually moves upward, and may be a little disturbing, but it makes its way up to the pharynx eventually, where it's swallowed. And this is a very adaptive mechanism to move bacteria, for instance, that are inhaled, and dust particles into your upper respiratory tract, and frankly, just to clear tiny particles. Imagine yourself, we've all, at times we breathe dust and smoke and all sorts of particles, and so your lungs are pretty good at getting most of this out from deep down inside the lungs, because of course the worst thing that can happen is that it becomes embedded down there, and that leads to changes in your lung tissue, some of which we'll talk about later in our class. So this mucus escalator is the mechanism by which your lungs prevent things from getting trapped. Now, things can go wrong with that mucus escalator, so we're all familiar with cystic fibrosis, and we're aware of the fact that cystic fibrosis is a mutation that's in a chloride ion channel transporter. And so, of course, what's supposed to happen is shown here on the left at the bottom of our slide. Chloride ions are transported across the epithelial membrane, and water follows. And so this water provides this liquid, this slick substance upon which the mucus rides as these cilia beat, and it allows the mucus to move freely up these tubes to the pharynx where it's swallowed. In cystic fibrosis, this mutation prevents the transportation of these chloride ions, and therefore the water that this mucus is supposed to ride on doesn't form. It doesn't end up on top of those epithelial cells, and so you have this buildup of mucus in the lungs, which is problematic in and of itself. But you also, aside from just the accumulation of mucus, you're not getting rid of all of these particles that you're supposed to be getting rid of. And so treatment for cystic fibrosis typically involves the removal, as you know, on a daily basis of this mucus via a percussion vest, and preventative treatment with antibiotics. It's a disturbing topic. It's the most common lethal genetic disorder that occurs in the United States or in most populations in the world, as your book describes. But it frequently necessitates a lung transplant and has a significant impact on overall survival. We make our way now to a discussion of the organization of the respiratory system at a microscopic level, all the way down there in the alveoli themselves. And so if we look at the alveoli, the arrangement of those alveoli, then there are two things that we want to take a look at here. So this picture from our text shows the alveoli themselves. Here's a single alveolus. This is another alveolus. This is another. This is another. And so they're clustered together here, as you can see. And then you can also see the blood vessels, the capillaries, making their way through there. So capillary, 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 capillary. And then B of this diagram is perhaps even more useful. And so here we see the wall of a single alveolus. We have alveolar air here. We have a capillary here, a capillary here. And we see that there's not too much to these structures here. The walls are very thin, which of course allows the exchange of oxygen and carbon dioxide between air and blood. So all that these structures consist of is the following. We have a alveolar endothelial cell, and these cells are referred to as type 1 cells. So what I'm pointing to here is a type 1 cell. This is a type 1 cell. This is a type 1 cell. Most of the endothelial cells within the alveolus are type 1 cells. The type 1 cells make up most of the inner portion, the inner walls of the alveolus. There are also type 2 cells that are present. The type 2 cells, the purpose of them is to secrete surfactant. Surfactant reduces the surface tension, and we'll talk a great deal about surfactant as we move forward. But most cells are type 1 in the alveolus, but type 2 cells are present to release surfactant. Then we see the capillary, pulmonary capillary. There's nothing surprising about the capillary. The structure is the same as we discussed in Chapter 12. But what we do notice is that we have a basement membrane for the capillary, a basement membrane for the alveolar type 1 and type 2 cells. We have an interstitial space here that's filled with interstitial fluid. So diffusion of oxygen and CO2 does make its way, of course, from here in the air to here in the blood. We do notice that the space of the interstitial space is quite narrow in places, such as right here, and in other places it's totally absent, such as right here. And in this case, the basement membrane between the capillary and the alveolar cell is shared. And so there is no interstitial space, and so there's even less room for the gases to make their way through as they go from air to blood or vice versa. Over here we have another nice picture. This is from an anatomy text. We have our type 2 alveolar cell, we have our type 1 alveolar cell. We have the surfactant, which is being shown in this little blue line that we see there, lining the insides of the alveoli, which is necessary for us to breathe, and for any length of time anyways, we'll talk about shortly. And then we also have a macrophage pictured here, which is not pictured in the diagram from our text. And so macrophages are there to mop up any pathogens that make their way past that ciliary escalator all the way down into the alveoli. Before we talk about the mechanics of ventilation, we want to talk briefly about the anatomy of the lung so that we'll have sufficient understanding to talk about how inhalation and exhalation occur. So let's take a look at the diagram at the bottom of this page, which gives us a brief overview of all the different parts we'll talk about over the next couple of minutes. We, of course, see the trachea, we see the bronchi, and we see the lungs. And furthermore, we see that coating each lung is a very thin layer of tissue known as pleura. The pleura that is stuck to the lungs and connected via connective tissue very closely to the surface of the lungs is known as visceral pleura. So what I'm running the cursor over right now is definitely visceral pleura. If we look at the inside of the thoracic cavity, we see another identical, aside from location, pleural layer. This pleura is known as parietal pleura. So the parietal pleura lines the inside of the thoracic cavity, the visceral pleura lines the lungs. In between those, we have a very small amount of fluid. The total volume of this fluid is only a few milliliters, and it's misleading, a bit misleading in this particular diagram because we have a wide space that is shown between the parietal pleura and the visceral pleura. In reality, there's almost no space between the visceral and parietal pleura. Now, I know I just said there's intrapleural fluid there, and that is true, but not much. We said there's, the thoracic cavity is a fairly large cavity, and a couple milliliters is not much fluid at all. And so it's described more as a potential space that exists between the visceral and the parietal pleura. There's a negative pressure there, interestingly. So it's like it's been vacuum sealed. And that's necessary, as you may realize, and as we'll talk about momentarily, for ventilation to occur. In other words, for the lungs to inflate and then to deflate. Your book describes it much like two microscope slides that are stuck together. And so we've all dealt with microscope slides and tried to separate two microscope slides or maybe two cover slips. And you know that if those two cover slips or microscope slides are wet, it's very difficult to pull them apart. And so this is the same idea. We have this negative pressure, the two surfaces are stuck very closely together, and so they are stuck to one another. They're not physically attached, but that negative pressure binds them together, and that fluid prevents any type of friction from occurring between the visceral and the parietal pleura.